The North Atlantic Library System acknowledges that the lands on which we live and gather are the appropriated homelands of Indigenous peoples. We want to express our deepest respect to those peoples past and present, including the Hoh Tribe, Jamestown Squamish Tribe, Lower Elwha Clown Tribe, Makah Indian Tribe, Quileute Tribe, Quinault Indian Nation, Port Gamble Squamish Tribe, and the Squamish Tribe for their care of these lands throughout the generations. Let us learn more about the histories, cultures, and traditions of Indigenous peoples. Let us strengthen relationships with sovereign tribal nations to provide an inclusive space fostering innovation and collaboration at the library. Because we have a slightly different setup tonight with our presenter online, if you are here in person, we will again hold all questions to the end. And if you have a question, you can raise your hand and we will bring the microphone to you. Otherwise, our presenter will not hear you. For our folks online, if you would please keep your microphones off and your cameras off so we can make sure we have as much bandwidth for our presenter as possible. You can put your questions into the chat and Korena will be able to see them or I will read them as time allows, but that way there aren't people popcorning in. So questions at the end. Okay, thank you. I'll turn it over to Dean.
Thank you. Good evening and welcome to our Perspective series. My name is Dean Butterworth. I'm the Parks Outreach and Education Specialist. So thanks for braving the cold weather and the icy roads to come out here and join us in person. We've kind of got a unique event tonight. We haven't done this before where our speaker is going to be virtual. One of the things that came out of COVID is we figured out how to do things virtually. So this is great. So our speaker, Korena Mafune, is at the University of Washington, and she's able to join us virtually and not have to deal with ice and roads and weather and everything like that in February. So it's great that she's able to join us. And we still get to enjoy community here together in the room as a group. So please enjoy the refreshments that have been provided by the Friends of Olympic National Park, and we thank them for their support. We thank the library for setting up the room and making sure all of the technology works. And we also thank the Parks Cooperating Association, Discover Your Northwest, who provides funding for the speaker's honorarium. And without everyone working together, we wouldn't be able to offer this series. One of the things that I like to think about perspectives is that there's lots of different ways of engaging with and enjoying and utilizing Olympic National Park. And one thing that we don't necessarily always think about is the fact that the park can be used as a laboratory and as a research space. And so these speaker series really focus on science in the parks. And so we're really fortunate today to have with us Korena Mafune, and I will turn it back to you, Korena. Thank you for joining us today.
All right, sorry with the laser pointer. It was a little weird to get my audio back on. Yeah, thank you so much, Dean. And I really always try to make it out for these things, so I'm sorry I can't be with you today. I also love the sense of community and being able to face to face get excited about science with other people. So do please forgive me. Dean knows that I was struggling with not being able to be there in person tonight. So I'm very excited for everybody to come together, whether it's on the computer or in the room, to listen to me talk about one of my passion projects. Also, it gave me a PhD, so I should probably be a little thankful for that. But overall, I'm going to talk to you guys today about canopy soils in the old growth ecosystems. So I always let me figure out how I can get to the next slide. There we go. I always like to start with acknowledgements. Sometimes people leave them to the end, but I think that it's pertinent to this research and this huge project that has spanned over a decade now, to give everybody a little bit of credit before we go into the projects, because you will hear me say their names throughout. You will also see some images taken by wonderful people. Any amazing photos you see were probably not taken by me. So, First of all, I would like to thank the group that has helped me in the field the most. We have Ann Polyakov, Lizzie Stone, and Alec Mead. They have all since graduated with undergraduate degrees, master's degrees, and PhDs and are on to new things, but we still stay in close contact. I also want to credit Irene Yee. She is a amazing photographer. So if you like some of these pictures, I always say support your local artists. Same thing for Kevin Henze. He took a lot of photos as well. And then, of course, I have to thank my PhD supervisors and my close friends who I still collaborate with to this day, Dr. Dan and Christina Vogt. There's Lizzie again. And then I also want to thank Michael Warner. He is a phenomenal nature director. He's done some work with me out here for science communication. And then contrary to popular belief after today, I do actually do a bunch of other research in a very large lab that I'm very thankful for. So I also want to credit Dr. Mari Winkler, who, while we do our research together, she also allows me to still collaborate and conduct research out here. And now she is interested in getting involved in research out here. So it really takes a community to make things go around. I'm not going to just credit myself throughout this talk. So the way that I really like to go into discussing my research is to first kind of give a crash course on fungal organisms. If I went right into this presentation just talking about fungi, if you're a fungal enthusiast or a mycologist of some sort, you may get some of these terms. But I do believe that kind of collectively explaining the ecology of fungi will really help bring this story together as we go through some of the more heavy data slides. This was the first slide that I talked about that you didn't see. We have Anne, Lizzie, and Alec. Irene Yee is a phenomenal photographer. Support your local artists. Did it go to the next slide? Yeah.
Yeah.
Okay. This is Dr. Dan and Christina Vogt. They were my PhD supervisors. They are amazing individuals who still advocate for old growth forest conservation to this day. This is Michael Warner. He is a nature producer, and he has taken some phenomenal footage out here with me, so I'd like to credit him. And then this was the lab that I'm in currently. We do a lot of research on bacterial fungal interactions, and now Mari and I are working together to expand research on the old growth forest together. So kind of a full circle acknowledgement. All right. So what I was saying when you guys were just staring at one slide, wondering what I was talking about, was that when fungal organisms first evolved on the terrestrial landscape, so on land from the aquatic environments, there is evidence that shows that fungal associations were vital to the establishment of plants on land. And what has happened throughout this divergence? So, when plants first evolved on land, they were bryophytes. By bryophytes, I mean very simple plants that don't have vascular tissue. So, we're talking about mosses, hornworts, and liverworts. We have a large diversity out in our rainforests. But then, as things continue to evolve into more complex plants, for example, lycopods, ferns, conifers, and then flowering plants, so angiosperms, what stays relevant? Why is this? Okay. What stays relevant is that throughout this evolution of plants on land, about 85% of plant families have sustained this mutualistic relationship that fossil evidence shows us existed back when plants evolved on land hundreds and hundreds of million years ago. So I just want to start with that big stamp of emphasis that fungal organisms were vital to land plant evolution. When you see plants, you often see the above half of plants, but there is a whole below half that I really hope to get people excited about today if you weren't already excited about it. So when we think of fungal organisms, I'm an ecologist. So I always think about not only their role in ecosystems, but how it also affects other organisms within that ecosystem and also other ecosystem processes. So, for example, how do fungi influence nutrient cycling? How do fungi benefit plant growth? How do fungal organisms potentially cause a detriment to an ecosystem, for example, through pathogenicity? So, as we go through these roles, I'm going to simplify it, but overall, I think it's really important that we first understand that there are fungal decomposers. So, this is one main group of fungal organisms, again, thousands of species that are fungal decomposers. So when we look at fungal decomposers, we have white rot fungal organisms. And this is not going to be anything where it's like you need to remember that there's white rot fungi and brown rot fungi. I'm more so listing out these parts to just highlight how powerful these fungal organisms are, because white rot fungi are lignin degraders. So lignin. It is an extremely complex compound that makes up the majority of woody tissues, and it's also found in leafy tissues as well. Fungal organisms are the only group of organisms on this planet that have evolved to be efficient in the breakdown of lignin. So when we think about how things get recycled to the ecosystem, how these giant old growth logs get broken down, first, fungal organisms come in, they excrete enzymes, help break it down. There are a lot of other organisms that also go in and help break it down too, but they cannot break down lignin like fungal organisms do. So there's also brown rot fungi. They can break down cellulose. So that is another very common compound within plant tissue. There's also lignocellulose, so lignin, cellulose, all of these complex structures that we're not going to get into today because that's not what we're here for. But overall, I just like to emphasize that fungal organisms are our world decomposers. They break down nutrients. They are our global recyclers. So they have a very key importance in ecosystem function. There's also soft rot fungi. Again, I could teach a whole quarter on these organisms, but it's just showing you the level of complexity when you start looking at these fungal groups. This laser. All right. So for example, something that you might see in your ecosystems or just in our backyards are turkey tail mushrooms. So turkey tails, they are a mushroom, but what I want to highlight is that turkey tails are just the fruiting structure of the fungus. It is a white rot fungus, so it is breaking down lignin, which is making up this log here. And so what I want you to just take a snapshot of is if you let's think about, Miss Frizzle and the Magic School Bus. If you were to jump into the school bus that shrunk down and went into this log, you would actually be going in between all of the fungal hyphae. So that is the fungal organism that is in the log. So the fungal hyphae is established in this log, breaking it down, and seeing this mushroom grow all along this log tells me that the fungi is very productive in that log because it's fruiting the fungus. Also, when you see something like this, just keep in mind that there is a large majority of fungal species that will never form a mushroom. That is only a certain group of fungi. It's the Dicaria clades that form mushrooms. So there's other organisms in there that we would never even see to the human eye. So we talked about decomposers. I want to also pay some credit to pathogens. So I think that one of the things that it's not necessarily a bad thing, but to me, when you say fungus to somebody, usually the response I'll get is like, oh, fungus on your foot, or oh, fungal pathogens, extremely important. But also there's pathogens in ecosystems. They are also kind of the bad guys, but they are crucial in how ecosystems function over time. So no matter what ecosystem you walk into, there will always be pathogens. They can come in the form of root rots, heart rots, blights, rust. And the health of an ecosystem is a partial determinant of how pathogens establish. So, for example, our old growth forests in the Olympic National Park, those forests are very temporally stable. Yes, they have pathogens, but are pathogens wiping them out? Currently, not right now. Hopefully they will not. But one of the reasons that is because these forests have succeeded into old forests that are very stable. If you go into an urban park, for example, or a parking lot that they plant trees in the little garden beds, those are not in a stable natural ecosystem. They're in a more of a disturbed ecosystem, so they would be more susceptible to pathogens, as would something that's a non-native plant being planted in this ecosystem. Again, it would be more susceptible. So pathogens are important. You will never walk into an ecosystem without a pathogen, but they can have devastating effects, especially when ecosystems get knocked out of equilibrium. So it's a balance. So another thing that's really interesting is that some pathogens are facultative. When I say facultative, I mean that depending on the environmental conditions, they can fulfill a different role. So a lot of pathogens start off on living species as weak pathogens, but then when a windstorm, for example, blows over that tree will fall, and then that pathogenic fungi will just start functioning as a decomposer fungi. So there's some of that going on too. So this is just some snapshots of pathogens you might see. Again, we're talking about old growth forests today. I also do work with farmlands and agriculture, so there's some pictures there as well. But this is something that has had a history of devastating Pacific Northwest forests. That's laminated root rot. This is a blight and a rust. Those typically are more prominent on herbaceous plants, so crop plants, grasses. That's a strawberry right there, and that's a blade of grass right there. And then also more relevant to our forests, we have heart rots. So fungus that can grow up into a tree and hollow out the center of the tree. We also have fungal cankers, which can also harm the tree. So again, pathogens are present. When you go into an old growth forest, it's very unlikely that all of the trees in there don't have some sort of, weak heart rot going on. I kind of put it in the comparison of, would you live to be 105 years old and not have like hurt joints in your hand? Most trees won't live to be 200, 300 years old without some sort of pathogen interaction. But because they're in a healthy forest and because they've succeeded into that old growth stage, it does not hurt them as much. Actually, pathogens are very important, for example, for northern spotted owl habitat, because they hollow out trees and then the wind will blow the trees over and they will provide that snag for the habitat. So again, I like to credit all groups of fungal ecology. So now we're going to get into the group that I study the most and biasly think is the coolest. All fungi are cool. So there are also mutualists. So I just did a short crash course on decomposers, so our ecosystem recyclers, pathogens, kind of the ecosystem facilitators of succession. And then now we're going to talk about mutualists. I'm going to focus mostly on mycorrhizal fungi, but I do want to make just a clear statement that not all mutualistic fungi are mycorrhiza. Mycorrhiza is a term that's used to describe the association of a fungus with a root. So, the structures are really what define mycorrhiza because there are also other root endophytes. So endophytes mean a fungus that can go inside a root that can also benefit the root but don't form mycorrhizal structures. So again, now we're kind of getting into that like, oh, crazy scientists, like so much. We're not going to go there. We're just going to kind of talk through the different mycorrhizal types because this will become relevant. So mycorrhiza go into the tiny roots of plants and they form structure. So think if we sliced a root like a carrot. This is the carrot slice right here, very small, like a pencil tip. So different types of mycorrhiza associate with different types of plants. If we reflect back to the evolution slide, these are the evolutionary pathways that fungi have diverged over time, and they've diverged many times. So if we look back at early evolutionary land plants, arbuscular mycorrhizal structures are most similar to what they were finding in land plants. And to this day, arbuscular mycorrhiza associate with about 80 to 85% of plant families globally. So this isn't just like something to shake off, like, oh, like some plants have a mycorrhizal associate. No, like if you include ectomycorrhiza and the other types of mycorrhiza, about 85 to 90% of plant families globally have mycorrhizal associations. So in lieu of time, the two main mycorrhizal groups that you'll often hear focused on are ectomycorrhiza and arbuscular mycorrhiza. So I already mentioned arbuscular mycorrhiza is that early evolutionary fungal group that associates with a broad range of plants globally. So ectomycorrhiza is a little different. So if we look at the way that it forms structures in the roots, arbuscular mycorrhiza actually goes inside the roots, and then these are called cortical cells. They actually form structures inside the cortical cells. We'll go a little bit more into that just because it's relevant to the rest of the presentation. The comparison is ectomycorrhiza. They actually form what is called a sheath on the outside of the root, and then they go into the epidermal cellular layer, which is this first layer right here. And then they can kind of go in between the cortex cells, but they do not go inside the cells like our muscular mycorrhiza do. But in both cases of mycorrhizal ecology, these species have evolved over time to be able to exchange nutrients for carbon. So if we look at decomposers, fungi cannot make their own food. They're like us. They need to get their food from other resources. And fun fact, we're actually more closely related to kingdom fungi than we are kingdom plantae. So if you think about it, they're like our distant times like a million cousins. So, fungal organisms, decomposers, they get their food source from breaking down dead organic matter. Pathogens break down live organic matter. Mycorrhiza exchange carbon with live plants for nutrients. So think of it this way. Plant roots can only reach out so far. They also can be hindered in their growth. For example, if they come across a rock. Mycorrhizal fungi have much smaller mycelium. If you compare the size of mycelium to roots, they're like tiny little threads that can weave in and out of soils and go very far distances and access nutrient pockets that the tree may not be able or the plant may not be able to access itself. So the reason why I bring up ectomycorrhiza is one, I think that I'm probably around ectomycorrhizal enthusiasts. I myself am one too. But that's because ectomycorrhiza only associate with about 5% of plant families globally, but most of the plant families that they associate with are tall, woody trees. What do we have in our backyard? A lot of tall woody trees. So we have a very high diversity of ectomycorrhizal species that associate with trees around here. We also have a very high diversity of arbuscular mycorrhiza. These other mycorrhizas, they get a bit more complicated. This is also not the end to the mycorrhizal continuum. So if you're excited about that, please reach out to me. I'd love to talk to you more about it. But the last thing I'll point out before focusing a little bit more on arbuscular mycorrhiza is that orchid mycorrhiza are a certain group of mycorrhiza. Orchids are one of the most diverse plant families globally. There's like 20,000 plus species or species in the orchid family. A majority of orchids would never germinate, which means they would never grow out of their seed state without a mycorrhizal partner. So orchid mycorrhiza is its own group of mycorrhiza, and orchid seeds actually will have mycorrhiza in that seed that help the orchids get the nutrients they need to grow in their harsh environments, because a lot of orchids are epiphytes, which means plants that grow on plants. So they can't really get into as much soil, for example, as something that was growing on the ground. Anyway, you will see that I get excited about fungi and then I'll start nerding out and going on a tangent, and I'm going to try not to do that. So, as we go into the details about arbuscular mycorrhiza, and you might be saying, why does she keep talking about arbuscular mycorrhiza and not ectomycorrhiza? Is because arbuscular mycorrhiza is the associate with the tree that we're going to talk about in a little bit. But before we get there, I just want to shed light on some of our Pacific Northwest ectomycorrhizal species. For example, we have our porcini or penny bun, boletus. There's a lot of different boletes that grow around here. To scientific knowledge, they are mostly all ectomycorrhizal. There's some arguments about, ones that are a little more decomposers, but typically they're considered ectomycorrhizal. So an ectomycorrhizal species that we'll see. So if you see a bolete popping up, you know that it's associating with one of the trees that is surrounded. And then a couple of my other favorite ectomycorrhizal mushroom species. So ectomycorrhiza, if I didn't say this on the last slide, a good amount of ectomycorrhiza fruit mushrooms as the reproductive structure. So again, mushrooms are like an apple to an apple tree, except they are the reproductive fruit of a fungus. Apples are the reproductive fruit of the tree. These mushrooms, they hold spores inside their structures. So that's the actual reproductive organism of the fungi, kind of similar to a seed, but it's not a seed. So I don't want to say it's like the seed of the fungus. It is the spore of the fungus. Also, before plants evolved seeds, they had spores, and you can see that with ferns and stuff. So anyway, what came first, the spore or the seed? Definitely the spore. Anyway, so we have our golden chanterelle. We're also really lucky because we have several different chanterelles around. We have the white chanterelle as well. So chanterelles are also ectomycorrhizal species. Amanita muscaria is also ectomycorrhizal. So are all of the Amanita species in the Amanita genus. So, for example, the death cap, Amanita phalloides, Amanita pachycolia, those are all ectomycorrhizal mushrooms. So I always just like to share the beauty of ectomycorrhiza. For example, there are also pathogens and decomposers that can form mushrooms as their reproductive structure. But again, I want to reel back and really emphasize that we're not actually sure about the percentage because there's so many fungi that remain undiscovered, but I've heard estimates that between only about 2 to 15% of fungal diversity fruits mushrooms. So most of fungal diversity remains elusive in its state for all of its life cycle, which are like these little ones that I study. So this is our buscular mycorrhiza. So our buscular mycorrhiza associates with roots a bit different and it reproduces differently too. So it is not as closely related to the mushroom forming species as they are to each other. So what our buscular mycorrhiza does is, again, it goes into that root as we talked about a little bit earlier, and then it forms spores underground. So these spores, unless you sift through soil like me and look under a microscope and get really excited, you will probably never see them. But they are traveling around on your shoes. They piggyback on slugs and snails. They can actually survive in a bird's gut all the way from one area of the world to another area of the world. So they are very small, but they're also very beautiful in their nature. They have like little lipids in them that you can see. Again, I'm not going to go off on that tangent. So, what do we want to look at when we start looking at species that associate with mycorrhiza types, especially our buscular mycorrhiza? Is we want to look at the structures that are in the roots. So, this is a educational photo that a friend of mine drew. And I honestly think it's the best depiction of our buscular mycorrhiza in a root, not because they're my friend, but because it's really accurate. So our buscular mycorrhizas, they can, and then if you look at this picture over here, this root, like that's the size of root that we're talking about. So we're at a very, very small scale right now. So when you go inside those tiny roots, so there's these structures that are called arbuscules. Those are the structures as well as these coils that will exchange nutrients with a plant in exchange for carbon. So again, plants can photosynthesize. They take in the CO2, they break it down to sugars. If they can have excess sugars, then they can distribute it to their mycorrhizal fungi in return for the nutrients that the mycorrhiza uptake. They can also uptake water as well for the plant. So there's a couple other structures that happen within these roots. There's vesicles, which are storage structures for the fungus. And then there are just different branching hyphae throughout the entire root. And so when they go into the root and they start coiling or forming these structures, that really denotes that there is nutrient exchange going on. If you were to look at a root and there were only vesicles, that might mean that there's some interesting thing going on where there might not be that much exchange because it looks like the fungus is just kind of going into a reserve mode. So again, that might be way too much in detail than you wanted, but you're here with me for the hour, so I get to tell you it. So, what really sparked my interest in research and why I'm, still looking at this stuff today is because you can zoom in even further. So we were just talking about fungal hyphae in a root, right? So when you have a soil system that doesn't have mycorrhiza, there is all of this excess space that is not being exploited or utilized for resources. When you have a mycorrhizal association, you have what they call this mycorrhizosphere. And then you can even zoom in further. So this is a piece of fungal hyphae. This is called the hyphosphere. And these are tiny bacteria that are huddling around the hyphae. And there's a lot of research going on right now that I am part of that looks at how these bacterial organisms interact in a beneficial way with these fungus that interact in a beneficial way with this plant. So how do bacteria help fungus, help the plant, help each other? It's very complex and very exciting. All right. So when we think about linking these relationships and interactions to ecosystem function, hopefully I have planted the seed, pun intended, that it gets really complicated, especially when you try to go below ground and really look at things that are not very visible. And hopefully, I also have been able to emphasize that fungi play a major role in the forests that we see out in the Olympic National Park, especially the old growth forests. So, for example, throughout all these different successional stages, there are fungi. When we have earlier tree species, we have more of a nitrogen fixing, AMF kind of EMF. Sorry, arbuscular mycorrhizal fungi is AMF. EMF is ectomycorrhizal fungi. So we'll have a mix of fungi, then we will get kind of a dominated arbuscular kind of establishment, then it will go back into mixed. And then if we have a lot of, for example, Doug fir or Sitka spruce or hemlock coming in, we'll kind of shift over to a ectomycorrhizal community. And then when we get into the old growth forests, I probably don't have to convince y'all because you're here listening to me talk about Olympic National Park, but we have some of the coolest forests. And that's not only because canopy soils, which I promise I'm going to get to, but it's also because these mixed forests are mixed dominated forests. And when I say mixed dominated, yes, I mean, in the sense of plants, but I mean in the sense of below ground fungal associates. So if you get really deep into mycorrhizal ecology, sometimes you'll hear people refer to forests as ectomycorrhizal dominated forests or arbuscular mycorrhizal dominated forests. For example, a lot of the Amazon rainforests are arbuscular mycorrhizal dominated. A lot of the boreal forests up in Canada are ectomycorrhizal dominated. But right here in the Pacific Northwest, we have a very diverse mix of both. For example, Sitka spruce, ectomycorrhizal. Bigleaf maple, arbuscular mycorrhizal. And then we have salmonberry, we have ferns. Ferns just do their own weird thing. I'm not even gonna get into that, but usually arbuscular mycorrhiza. And then salmonberry is arbuscular mycorrhiza, and then hemlock is ectomycorrhiza. You can probably get where I'm going right now. Our below ground ecosystem in these forests is amazing. So every time I walk into the forest, I start to zoom in and look very closely at the interactions that are happening because that's just kind of how I've trained my eye to work. And I think that's one of the most interesting parts. So, for example, if we go back to this slide, we have like an old log right back here. I get really excited. I run up to that log. What's on that log? Such a diversity of amazing species that we can see. We have lichen. For example, lichen are awesome. They're a composite organism. So, for example, lichen being composite means that they have a photosynthetic algal cell. So this is usually cyanobacteria. And then they have fungal hyphae that provides the algal cell a habitat to live in, and then they form the lichen. So a lichen is named after the fungus that creates it, but it's really a composite organism. And then also within this lichen, there's a whole nother community that's not just the algae and the fungi. So lichens are complex biological systems, and they're really cool if you're a lichen enthusiast. We have a great lichen diversity out in the Olympic National Park. And then, so for example, we have some mosses sprinkled in with this lichen too. Also, spike moss, which is not true moss, but it's all over the place. That's Solanginella. So, what if we zoom in even further? Oh, hey, there's that moss. Oh, is that a little mushroom that we see? Yeah, it is. That is the stuff that really gets me just appreciating the beauty of nature and the scales, like this beautiful, huge ecosystem. How does it exist? How does it function? And how did it get here? It's really these organisms underground or in the pockets of the areas that you can't really see that are facilitating this. And this, I would say, is probably about one of the smaller levels that we would be able to see. But if you were just walking through a forest pretty quickly, this would, I mean, moss for scale. So what happens when we zoom in and then we zoom back out to these beautiful trees that we have in our old growth forests? So this is a bigleaf maple. This is in the Queets River watershed, and you can just see this copious amount of moss and spike moss hanging off of the trees. There's also other epiphytes like licorice fern. I have even seen a Sitka spruce about 4 feet tall growing up in the canopy of a bigleaf maple. So the possibilities are endless up there, and they're only been researched for about 1981 was Nilini Nadkarni's science paper on canopy soils. Nilini Nadkarni is the pioneer of these canopy soil ecosystems, has done a lot of fundamental science that has opened the doors for other people. If you were interested in more of this work, she is actually hosting a Nat Geo event in Seattle in a couple weeks. So I highly recommend to look into that if you want to hear more from her. But so when we zoom in to these beautiful treetops, we get canopy soils. So, what are canopy soils? I think that when a lot of people travel from around the world to see what we have in our backyard, they are really drawn to this area because of these beautiful mossy mats. But what I'm drawn to is what's hiding underneath those beautiful mossy mats. So canopy soils are thick mats of organic matter that are on these tree branches. And how they form is these are old growth forests, right? That maple that I'm in, you can see me right here for scale, is probably over 250 years old. Maples are also really hard to core, and also when you have trees that might be slightly decayed in their inner trunk, it gets even harder to core them. So don't ask me the exact age of this maple, but we have estimates between 200, a little over 200 years old. So, basically, what happens is as these trees get really old, the mosses start establishing on the tree branches. Every season, the leaves fall with the conifers, so the evergreen, Sitka spruce, they just kind of shed their needles throughout the year, which also land on there. So they intercept a lot of litter fall that decomposes over time, again, by fungal organisms, and then also other organisms like shredders and grazers. So insects, and then also bacteria have a huge role in decomposition. And they are prevalent in our old growth forests. So if you walk into an old growth lowland, especially ecosystem in Washington, just beautiful drapery. And so when I was thinking about researching canopy soils, I really wanted to know what they provided. So, some of the fundamental things that we know about canopy soils is they provide a habitat to a diversity of species. For example, this is a beautiful beetle with an earthworm about 20 feet above the ground. For a while there, I was wondering how earthworms were getting up there. I think this answered it for me, and birds can drop them. I don't think there's a scientific paper that claims how they get up there, but again, this is my theory. And so there's just a plethora of life. For example, canopy soils, especially in the larger conifer trees, are crucial habitat to the marbled murrelet, which is a cute little seabird that floats in the water most of the year. But I'm not a bird person, so I'm sorry if I'm offending any bird people with my terminology. But it actually will fly into the old growth forest canopies and it will nest there. It's one of the only bird species that both the mother and father nest the egg. They only lay usually one egg a year if they even do lay an egg, and their nests are literally just little circles of kind of poop that they make while they turn on these canopy soil mats. So, again, just crucial habitat. Mostly they've been studied for habitat for things that we can see, but I was really interested in what else they provided a habitat for. So, for example, here you can see this huge branch, and then I dug a hole in it, and then you can see these. So, wait, what's going on here? Because roots are supposed to happen on the forest floor, right? So, when I first climbed into these trees, it was with a PhD student. I was just a wee little undergrad, super excited about climbing trees. And what Camilla had found through her research was that these canopy soils in bigleaf maple and Sitka spruce, I'm just going to be focusing on bigleaf maple today, they actually enhance the carbon and nitrogen pools of these forests by 25 and 20%. So when we look at nutrient cycles, canopy soils are contributing a pretty large input to these ecosystems. Sorry, I keep getting little things on the screen. And then so the other thing that I was interested in was that, okay, so they have nutrient pools up there and they have roots. Again, we'll get to these. So these are adventitious roots. These are actually large. So for scale, those are probably about the size of like my finger. But what is really amazing is that they branch into these really fine root systems. And so when I first climbed up into a tree and I saw these roots, I was already excited about fungal organisms. So I was like, there's roots up here? What? And then Camilla was like, yeah, there's roots. And then I was like, Are they associating with fungi? When I first found out that Nilini had seen that these maple roots had fungal structures in them, I was like, okay, well, what does that mean? What does that mean for the forest resiliency? Like, if these canopy soils have nutrient pools, then these fungi could be pulling nutrients from their mycorrhizal associates, like in the canopy. Also, there's other fungal organisms that would maybe be associating with these roots that could also benefit them in times of stress. And why I say times of stress is because canopy soils are basically like a sponge. So when it's really wet, they are super soaked with water. When it gets really dry, they will dry out a lot faster than the soils on the ground because they don't have as much insulation. They have a lot more space in them because they're organic matter. And so when I found out there was fungi associating with them, first of all, I got really excited. I did that little dance down there. Then I started really thinking, wait, okay, but if there's fungi up there, what does that mean for the competition on the forest floor and also the competition in the canopy with the epiphytes? Like, are these fungi cooperating? Are they competing? What are they really doing? And then I started a PhD, and that's what I looked like through most of it. And we came out with some really amazing findings. So, for my PhD, what we wanted to look at was these root associated fungal communities in the canopy soil. And one thing that I want to break apart real quick is root associated fungal communities and soil fungal communities are considered two different things. So, I actually went in, I cleaned off the surface of these roots and extracted fungal DNA directly from inside the root rather than from the soil. And I say that because I'm going to talk about that a little later. So we wanted to determine the fungal establishment in these roots because that could tell us, for example, are we seeing as much mycorrhizal association in the canopy compared to the forest floor? That could give us some insights about how much these trees may be investing in their canopy mycorrhizal relationships. Also, do these communities and their diversity shift over time and space? Because if they're different between different sites and they shift seasonally, that could also give us more information about what's happening in these forests. And this, if you really want to nerd out, is actually published in Mycologia. So you can go and read some nitty gritty science. If you can't access this article, email me, I'll send it to you. So the two sites that we did this research in were in the Hoe and the Queets. You can see that they are spatially pretty aligned in the sense of the lateral location. You do have to drive out on a beautiful drive. Ugh, so hard to do along the coast. And then you drive into the Queets. So these two forests had very similar species composition. They were similar in age, and they were both along a riparian zone. So one was along the Hoe River, and the other one was along the Queets River. Sorry, I got another little ping. Okay, so I like to just kind of take us through a story of wandering into my field site. So when we wander into the Queets, we actually come across this beautiful legacy pasture land, which the trees have never encroached on, and the elk like to hang out there for their little safety space. So a lot of the times we would go say hi to the elk and then go into these beautiful old growth stands. So you can see me here climbing up into the tree to get those samples. So my PhD consisted of a lot of field visits, a lot of sampling, and then, of course, a lot of lab work on top of that. So when we looked at fungal colonization, so here, this blue color, that is a vesicle. You can see that there's arbuscules or more kind of coils right here. We talked about arbuscules and coils earlier on. This is why I wanted to kind of visit that morphology. We could see arbuscules over here in this black-white contrast. We can see a vesicle. We can see fungal hyphae running through. And so when we went in and looked at these fungal structures and actually calculated the percent of fungal structures, so for example, percent of fungi versus percent of no fungi in the root, we actually found my hypothesis that was that canopy soils would not have as much fungal establishment because there's dispersal limitations, right? So things cannot necessarily get up to the canopy as easily as they can be spread around the forest floor level. But this is one of the things where it's like, yes, my hypothesis was wrong. Canopy soils had very comparable amounts of fungal colonization in their adventitious canopy roots, which we can see here. So this is a violin plot. And again, I'm not going to focus too much on plots. I'm super happy to answer questions afterwards. But in brief, this plot is a violin plot with a box plot. You can see all the data points here. The purple ones are from the Queet site. The green ones are from the host site. We have the canopy. So you can see that there is more of a breadth of colonization percent across the samples than the forest floor area. But what you can see is if I showed you all the statistics and lined everything up again, which are in that paper that was in a previous slide, there is no significant difference between these two averages. So that means that the canopy roots are sustaining similar mycorrhizal associations in the sense of colonization in the root compared to the forest floor. So that was really exciting. These are just some really fun pictures. So let's think that we're slicing a carrot again. So this is the cross section of a root. There are so many fungal structures, which are these really dense green coils. So you can kind of see the cortical cell here. And then all of that green, that's just fungi everywhere. You know me, I love fungi. So when we started looking, this is a lateral section of the root. You can see fungi going in and out between all of those cells. You can actually see, so these blue outlines right here are the root cells. This is the edge of the root. You can see the fungi entering the root and coiling in the cells and going down and coiling more. This is now getting a little deeper into the root tissue, so it's getting a little harder to see. But the overall takeaway is there were fungal structures everywhere in the canopy roots. They did tend to be more of the coil type of arbuscular mycorrhizal structures, which again, if I had a whole quarter to lecture you, I would tell you the difference between what that means, and nobody really knows. But there's an arum and paris continuum of arbuscular mycorrhiza. So we were seeing a lot of coils. We also saw arbuscules, so hard evidence that there is nutrient exchange going on with these roots. So when we wanted to look at the diversity, what we did was this is called a PCOA plot. And basically, if I went in and I sampled the fungal community of a root, and then I said, okay, there's 100 species in this community. And then we go over to this one and I say, okay, there's like 150 species in this community. And then we do a bunch of calculations and they say, okay, that's how different these communities are. It basically condenses each fungal community that I sampled into a point. So what we can see right here is there is a clear divide on this plot between the green, which is the forest floor root-associated fungal communities, and both the hoe and the Queet sites. No significant difference between fungal communities. We didn't really expect to see that because they are similar old growth forests in kind of similar spatial areas. So although there were some differences, they weren't significantly different. And you can see that by these being mixed together. However, when we go up to the canopy level, first of all, the canopy soil fungal root associated communities were extremely different than the forest floor communities. You can see that because they are not close together on this plot. And then further, outside of a couple outliers, which are these purple squares from the Queet site, there was actually a difference in the hoe canopy communities and then the Queet's canopy soil communities. So that also was a hypothesis of ours because the dispersion, the way that things are dispersed in each ecosystem varies a little bit. And what gets dispersed up there may or may not be random. We don't know. We would have to look into that further. So overall, we found that root-associated fungal community diversity and composition was dependent on the type of soil, so canopy versus forest floor, and it was dependent on the spatial location at the canopy level. So then what we also wanted to know is, okay, so we have mycorrhiza up there, and then Camilla also found out that they contributed to the nutrient pools of these forests. But what we didn't know is within those nutrient pools, what is available for the plant to uptake and how does this shift seasonally? So while we were determining these fungal communities, we also determined nitrogen and phosphorus mineralization. So mineralization is the process of organic, for example, nitrogen or phosphorus being converted into inorganic nitrogen. Immobilization is the opposite, where nitrogen or phosphorus that would be able to be available for plant uptake gets bound to something or gets converted to something or get tied up in microbial biomass. To put that, in condensed terms, Mineralization means nutrients are becoming available that the plant can uptake, and mobilization means they're not available. And we do have issues with nutrient limitations in our forests and different ecosystems. But what we actually found through running nutrients seasonally for a year in this ecosystem is that canopy soils, they mineralize more phosphate, ammonium, and nitrate compared to the forest floor. So if we look over here, what this graph says is purple is nitrogen and blue is phosphorus. And so in the canopy, if we had a kilogram of canopy soil, it has so much more nutrients available on a yearly basis for those adventitious roots, for those epiphytes, for things in the canopy to access than the forest floor does. So that would be expected. Canopy soils are highly organic in nature, and organic matter tends to have more nutrients. But that means that the canopies are kind of like this little snack box for these trees and the species that dwell up there, which is really cool to me anyway. Um, throughout the study, we found several species of interest. This is a mushroom growing up in the canopy. So you're like, but Korena, you've been talking about fungi that don't form mushrooms. Well, now we're going to segue into why I'm a fungal ecologist and I don't narrow down my studies to any individual part of the ecosystem, even if it is my favorite arbuscular mycorrhiza. So right here we have a Mycena species. Mycena are a very diverse group of what some mushroom enthusiasts will call little brown mushrooms or LBMs. They are widely decomposers. Some of them can be pathogenic, but mostly they have always been researched in their saprotrophic nature or decomposition nature. So, of course, when we look at the species that we found arbuscular mycorrhizal fungi. Um, and we found structures and we've done some further studies. So up until this point, there were definitely people who would rebuttal and be like, well, what if like there's just mycorrhiza established? Like it could be just vesicles. Maybe there's not an interaction going on. Nope. There is hardcore evidence that these adventitious roots have mycorrhizal associates that are exploiting the canopy soils for their nutrients and potentially their water. We would have to do more studies to really narrow in to what they are pulling out of the canopy soils. But now nobody can argue with me that they're, associating with fungi that are also accumulating resources for the host. Because people were arguing that up until maybe, 5-6 years ago. So I mentioned Mycena. Again, like I said, I see fungus and I get excited. So I described Mycena over here. We see Mycena all over in the organic canopy mats. It's expected, it's a decomposer. But one of the fungal associates that was in the highest abundance in my roots was Mycena. So what's going on with that? Because one, if Mycena was a mycorrhiza, it would be an ectomycorrhiza. Big leaf maples aren't ectomycorrhizal associates. They're arbuscular mycorrhizal associates. So still kind of weird. Maybe it's just hanging out there, but if it's just hanging out there, why would it be in such high abundance? So there has been literature recently coming out that in certain conditions, Mycena actually go into the root and can have beneficial impacts for the plant. They often call things like this the waiting room hypothesis. Again, if we think about evolution, ectomycorrhiza evolved from decomposer fungi. So it wouldn't be an ectomycorrhiza with maple, but I have some hypotheses that Mycena in these ecosystems, especially up in the canopy soils, are doing something beneficial at that root soil interface, or maybe even inside the root, but it wouldn't be considered a arbuscular mycorrhiza. Remember, I mentioned all mutualists are not necessarily mycorrhizas. Some other species that we found of high interest are dark septate endophytes. So for example, if we look at this root here, you see this pigmented fungi kind of wrapping around the root over here too. This is kind of like a pseudo sheath. So dark septate endophytes, for a while they were thought to either be pathogenic or just not researched, but over the last 10 years, a lot of research has come out that, are they mycorrhizal? Are they beneficial? For example, dark septate endophytes have been shown to help plants when they are under drought stress, which happens a lot in the canopy. Interesting. Also, Melanomyces bicolor, that is a fungus that can be an ectomycorrhiza with certain plants. It can be an ericoidal mycorrhiza with certain plants. It can do a bunch of things. It's facultative. we found it up there. What is it doing? We have to do more studies to know, but maybe something interesting. Same thing with some other facultative species. So, long story short, there's a lot of interesting things going on at the canopy soil level in these tiny little roots that you would maybe never even think existed up there. So I'm going to kind of go through these next several slides kind of fast. You can ask questions about them. I just want to be respectful of time to allow for questions. That wasn't the only study I did for my PhD. On top of that, we also went up to the canopy and we put these roof structures up there that divert half the rainfall off. So that was mimicking drier summers. And then we put irrigation structures on some that introduced about 2 times the amount of rainfall. So that is a mimicry for wetter winters because these ecosystems are experiencing wetter winters and drier summers. And I really want to know what that might mean for these canopy soil habitats. And then, of course, we had our controls at ambient condition. So what we found across, so now we're up just at the canopy level, looking at how rainfall impacted these canopy soil nutrient dynamics and fungal community dynamics. And so what we found is that under these different structures, root-associated fungi shift under drier and wetter conditions, and so do mineralization rates. So there's a lot of interesting things that should be further studied up there to really understand what that means for preserving and managing these old growth forests as we move into these more environmental extremes. Additionally, I want to thank one of my undergrads that I worked with. They constructed these leachate catchers to catch what was flowing through the canopy soils. And we actually found that all of these available nutrients that we were measuring up there leach to the forest floor. So canopy soils are kind of disconnected, but they're still very connected in the sense of whole forest function because nutrients can get to the forest floor from the canopy, and so can a lot of total organic carbon. And again, carbon is the basis for all life. So really seeing these dynamics were pretty amazing. But wait, you might be like, oh, they're leaking to the forest floor. But what happens? Are they leaking into the atmosphere? If any of you are excited as me about this, you might be asking yourself that question. If not, I'm going to answer it for you anyway. So, another person that I worked very closely with, Lizzie, who you'll see on the next slide, Lizzie measured canopy and forest floor soil CO2 fluxes, so carbon dioxide, a potent greenhouse gas, and then methane, even more of a potent greenhouse gas. These forests really play a large role in the sources and sinks of greenhouse gases. So what Lizzie actually found, there's Lizzie right there measuring with her chambers up in the canopy. So Lizzie actually found that just bigleaf maple canopy soils, so one thing I want to plug here is we didn't even measure this in Sitka spruce. Sitka spruce also have copious amounts of canopy soil. So there's a lot of things that we still need to answer out there. But in just bigleaf maple, it accounts for 4% of the CO2 source in these ecosystems and also contributes to 4% of the methane sink. So that means that they are sucking in methane, but they are emitting a little bit of CO2. However, methane's about 63 times more potent than CO2. So we have to start understanding ways that we can get enough data to balance what these budgets mean for this ecosystem. These little tiny graphs are just some of the moisture and temperature data. We found that these were mostly linked to moisture regimes. So again, those wetter and drier seasons are going to impact greenhouse gas emissions, and we don't even know what that means for the canopy soil level. So now we are kind of approaching time. I do want to give a big shout out. So, after that greenhouse gas question, we collaborated with a wonderful group over at Western Virginia University. So, this is Dr. Jenny Kane and this is Dr. Ember Morse. And what we did out there was we climbed several trees, not exactly the same trees for my PhD study, but we sampled at different branch heights. And what they found was that soil respiration, which means the amount of CO2 coming out based on microbial activity, was higher in the canopy. It didn't really significantly differ between heights, but What we did find was also at those different heights, bacterial and fungal diversity was highest on the ground and decreased with branch height. But what I want to emphasize there is it doesn't matter if you have a higher diversity on the ground if there's too much functional redundancy. Like there can be something there. It doesn't matter if it's there, if it's not doing anything like super beneficial. It could just be dormant. So what actually this tells us is that because we have higher respiration rates going up in the canopy, we actually have more of a functional community in the canopy in the terms of bacterial communities and fungal communities. And if you flash back to about 15 minutes ago, I mentioned that soil fungal communities and root associated communities are different. These were soil communities. So now we're looking at the broader community of bacteria and fungi going up from the branches of these different canopy ecosystems. And so, on top of that, I am also a National Geographic explorer, and they were amazing in supporting me to go out there and do a mushroom sampling. You might have thought that I was all for, fungi you can't see. No, I'm for all fungi. I love the mushroom ones too. So we actually collected over 100 specimens from the canopy soil. A lot of them were really interesting. A lot of mycena. This is a hygrosabe, which can be mycorrhizal and might be associating with like the moss tissue or one of the other epiphytes up there. That was one of the coolest ones to find. Um, and so overall, there is a diverse, functionally significant microscopic community that is dwelling in canopy soils, but we still have to do so much more research to know what these interactions mean. Also, Jenny told me that they found like a super rare nematode up there that it was not a bad nematode. It was just a ecological good nematode that breaks down and eats things that doesn't harm other things. But they found a nematode up there that hasn't been found or noted in like decades. And so when they reached out to their nematode experts, they were like, "What? You saw this? Like, where did it come from?" So there's probably a lot of diversity that's never even been discovered up in these canopy environments. So, to sum it up, soil microbial communities are more active in the canopy than they are on the ground. There was a lot of nematodes and microherbivores that were found in Jenny's study. So, she was also looking at microarthropods and things like that. And the increased community activity, and that kind of points to that there's potentially more trophic interactions between these unseen organisms in the canopy, which again their evidence was kind of pointing towards it being more related to soil nitrogen. So overall, hopefully I have totally derailed how you look at these systems because canopy soil should not be overlooked, actually underlooked, because they're above your head. This is why I like being in person because, then I could gauge if people are laughing at my stupid jokes or not, but now I'm just laughing at myself. But overall, canopy soils may be a resilience compartment for these old growth trees and the forests that they dwell in, especially in a changing climate. So there is an urge to really preserve and better understand and define these systems because every time we answer a new question, it literally leads to that is an ecosystem scale contribution. We need to learn more. So I'm not going to go over bacterial fungal interactions, but I do want to say that I showed you a lot of cool tree climbing pictures. However, it's not all fun in tree climbing. There are so many things that go on out there, staring at trees, shooting a giant slingshot, tangling myself up in rope. I always say that my PhD is actually in tying knots rather than science. So much rain, expensive equipment, tree hugging. There's a lot of that going on out there too. A lot of makeshift. Oh my gosh, it's raining way more than we thought, and my 3 pairs of pants already got soaked. What am I going to do for the rest of the weekend? And that being said, I really hope that you could hear me okay. I'm sorry if I wavered in and out, but I'm also happy to stick around and answer any questions y'all may have.
Okay, thank you, Selena. Um, so this has been recorded and it will be available on YouTube, usually by the end of the week. So especially towards that beginning part when we couldn't always hear, if you need to go back and check anything, it'll be on YouTube within a couple of days. And yeah, Okay, so if you have a question, here in person, you can raise your hand. Audra will bring you the microphone. If you are joining us online and you have a question, please put it in the chat. That way we can read it out so the people that are here can hear the question as well. One here, okay.
Hi, thank you so much for that. I was curious what the roots in the canopy, what plant the roots in the canopy belong to. The ones that were about a finger thick.
Yeah, sorry if that wasn't clear. I don't know if I missed that in my excitement of talking too fast or if I cut out when I maybe said it. I'm gonna go with the former, but hope for the latter. So the adventitious roots, they're coming out of the maple branch. So as the maple would grow roots into its floor, like a typical plant does, they're actually growing roots out of their branch tissue into those canopy soils. So when I would say things like for the host tree, I mean the big leaf maple is actually growing roots out into their canopy environment to also take resources from those nutrient-rich canopy soils. There's also roots from like fern rhizomes, and there's a lot of other epiphytes up there. So there are other roots, but these roots that we did for this analysis were big leaf maple adventitious roots belonging to the big giant trees.
Thank you. A question from online. Is there any way to determine the rate of increase of canopy soils?
Yeah, I try to get that funded, and science funding always depends on the atmosphere and the agencies and everything. We have not determined that yet. I have some theories, and I would like to determine how long it kind of takes these old growth forests to develop what we termed their canopy soil legacies. Um, so just with my experience in these forests and also, working in and out of other stands that are much younger, so like mature stands and things like that. So Sitka spruce are a later successional species, and the Sitka spruce will usually be growing for several hundred years before they kind of start thinning out. And then when they start thinning out, that's when the big leaf maples kind of take their opportunity to start growing in those little sun pockets. And then I think that maples love moss, so it's probably pretty soon after they kind of gain their structure that they have that moss layer. But I would hypothesize that, so to put this in perspective, um, I have been in canopy soil mats that are like 2 feet deep from the branch. Like these branches, some of them are actually pretty small and they look so big because of the canopy soil that's wrapping around them. And so if you have a giant mat of canopy soil that is like a foot deep on a branch, I would hypothesize that it takes over 100 years for it to get that thick. Hypothesis.
Any other question?
Well, as you're probably well aware, there's no shortage of clear cuts here in the peninsula. And so when the trees go back, how do the organisms recolonize in the canopy soil? I assume the fungus or spores are traveling aerially, but there's a lot of vertebrates there too. It seems like this would be very important for the health of the tree to get established quick.
Yeah, so to answer that question, I think that first of all, some of the fungi, especially ones that aren't mycorrhizal associates, so for example, some root endophytes can also be leaf endophytes. So when the leaves fall on the branches, the fungi that are in the leaves then have a chance to take foot in the canopy soil environment. And bacteria as well. So that's one of my hypotheses for how bacteria and like endophytic fungi get dispersed up there. My hypothesis for the arbuscular mycorrhiza is you would be so surprised how many times I have climbed up to like an 80-foot branch and I have seen a snail or a slug up there. And I'm like, how long did it take you to get here? But so yeah, they're fungivores. they, move through the soils, they can pick up things, and then they decide that they want to crawl 50 feet up a tree. So that's another potential for dispersal. And I think that I heard you also mention bird dispersal. That's probably one of the main ways that they also get up there, because birds will go in, they'll kind of pluck things out of the ground looking for food, depending on the bird. And then they'll go up in the tree and do stuff up there and all of that. So they call that like zoophagy, hydrophagy, things like that. Different ways that spores can get into the canopy environment. And I think that your line of thinking is directly in line with mine on, yeah, like bird dispersal. Even wind dispersal can have a effect depending on how disturbed the soils get in the wind event, or if like, a herd of elk run through and kind of toss things up. There might be some aerosol dispersal ways, but arbuscular mycorrhizal spores are actually large enough that some of them are too large to really be picked up heavily by wind throw, things like that. So there's a lot of interesting questions there that could be answered and have not. And I do agree that the establishment is important for these trees. But it also really reflects forest succession too. So there's so many complicated things that really go into making an old growth forest in the first place. And then so figuring out what it takes for canopy soils to establish an old growth forest is, a whole new layer of complexity, and that I'm excited to pursue, hopefully as a lifelong thing. Hopefully that answered your question. It was also going in and out a little bit, so I try to piece it together.
There's an interesting section in the book Gathering Moss by Robin Wall Kimmerer about the transmission of moss spores, and some research that they did with like squirrels and stuff, and slugs were the answer to that. So that was a fun book to read. I'm not sure if that would answer all of the question. One last question from our group online. Are canopy soils saturated often enough to be classified as histic soils?
I love your question. So I cannot take credit for this push, because this was actually a push that Nilini did back probably before I knew canopy soils existed. But so in the early 2000s, when there was a lot of canopy soil papers coming out, there were, of course, people coming out and saying, what do you mean a soil? It's on a tree branch. Like soils are like derived from minerals. And okay, as a soil scientist, false. There are organic soils. So, for example, there's mollisols, there's histosols. Those are organic soils on the ground. They're soil series. So, when these arguments started coming out, being like, they're not canopy soils, you can't call them canopy soils. There is a paper, Nilini in 2002. She actually put forward the term arboreal histosols. And then in 2006, I believe it was a group in the redwood forests that was doing canopy research that actually designated the term arboreal histosols in their publication. And then Camilla, who I was working with when I was an undergraduate, she actually defined the soil horizons in Sitka spruce and bigleaf maple. And so they have different, like bigleaf maple is a haplohemist histosol, and now we're getting really into soil nerdology. So sorry if I'm losing some interest in that, but yes, they are an arboreal histosol. They are categorized as a soil, and also they do derive, mineral deposits from the air and stuff. So I just wanted to note that too. I had a friend that was doing a study, and they actually found some isotopic tracers from like some pretty far wind throw. I think maybe from somewhere in South Asia, like they were able to trace it back. So things can travel pretty far.
Okay, thank you so much. We'll respect the author's time or the researcher's time. Thank you so much for your presentation, and we hope you all come back next month. Thank you.
Thank you guys. I really appreciate it. And I'm always so excited to answer questions and engage. So let me know if you have any further questions for me. And I also love that hi, everybody online that came in to say hi. Yeah, so when you mentioned that book, that's also a really good point, because so if you look at a lot of research, it says that mosses are the one bryophyte that do not form mycorrhizal associations. Liverworts, they're all over liverworts. None of those really have a true rooting system, but you can find mycorrhizal structures in the non-vascular tissue of the mosses and the other bryophytes that grow there. So I would say that's very in line with what you were saying. That's from that book, is also things can grab that moss, go up there and burrow. I don't know how many squirrel houses I've accidentally stumbled upon in my days up in those trees. So
Okay, thank you. Have a great night. And everybody drive safely on the way home. Careful. And we'll see you next time.
thank you.
Great.